A single bacterial colony can multiply into the billions overnight. When you are monitoring a culture, tracking this replication rate is mandatory. But at densities of millions per milliliter, the human eye cannot track individual cell membranes fast enough to produce a reliable count. The sheer volume and three-dimensional density of a liquid culture creates a bottleneck. Manual observation is limited by the physical speed of the researcher, creating a gap between the biology scale and our measurement. To bridge this gap, researchers engineer biological proxies. We move past looking at the cells and start looking at the signals they produce, converting their physical presence, their metabolic engines, or their chemical footprints into readable data. We are going to map the mechanics of cell quantification. By deconstructing physical, metabolic, and compositional assays across bacteria, animal, and plant models, we can see how measurements are scaled to match the population. Quantitative biology addresses the problem of scale by translating biological processes into measurable physical optics, such as light scattering, absorbance, and fluorescence. This image displays a Petroff-Hausser counting chamber, a foundational tool for direct physical counting. A microscopic grid allows a researcher to manually tally cells within a precise volume of 0.02 cubic millimeters. However, it is labor-intensive and error-prone due to uneven cell distribution. This schematic shows turbidometry. As incident light I hits the cuvette, cells deflect it. Applying the Beer-Lambert Law, we measure this light reduction to isolate concentration, C, calculating the optical density automatically. However, physical optical density contains a critical failure point. It strictly measures total suspended biomass. The light scatters off cell membranes whether the cell is functioning or completely inert, making it impossible to differentiate a thriving culture from dead cells. This diagram illustrates flow cytometry. It uses a fluidic system for hydrofocusing, forcing a dense suspension of cells into a strict single-file stream. As each single cell passes the targeted laser beam, a photodetector measures the specific diffraction event, logging a precise physical count. Flow cytometry achieves high physical precision by automating the count. Yet, without specialized fluorescent tags, it still fundamentally counts physical objects rather than confirming cellular health. To prove viability, the biological requirement changes. We have to stop measuring physical shapes and start measuring active metabolic engines. This shows the transformation of MTT dye, a biochemical tool used in animal tissue cultures. The yellow MTT crosses the membrane and interacts with active mitochondrial dehydrogenase. The enzyme reduces it into an insoluble purple compound called formazan. Here we see the reduction pathway for the resizurin assay, an alternative colorimetric proxy. Active dehydrogenase enzymes reduce resizurin, a non-fluorescent blue dye, into resirufin, which emits a bright red fluorescence. By linking a dye directly to a metabolic engine, we establish a mathematical rule. Optical absorbance readings, like measuring the purple formazan at 570 nanometers, are now strictly proportional to the number of viable cells. Dead cells don't process the dye. This diagram maps the equivalent viability proxy for bacterial cultures, the serial dilution and spread plate method. Researchers dilute the parent culture by factors of 10 until cells are scattered sparsely. After incubation, every visible colony proves the existence of exactly one viable dividing cell. By measuring active enzymes or confirming replication on agar, metabolic assays successfully decouple raw biomass from true viability. Researchers extract the actual health of the culture, bypassing the dead weight entirely. Monitoring microscopic plant tissues introduces a different mechanical problem. Plant cells are locked in rigid matrices. They cannot be easily suspended in a cuvette or hydrofocused through a laser. In this 3D wireframe animation, we see an imaging-based approach. Cameras map the coordinates of the plant over time, tracking phenotypic growth markers like stem height and leaf surface area. But to achieve precise cellular quantification, researchers pivot to a biochemical approach. They extract intracellular contents, specifically chlorophyll, to estimate growth based on the physical accumulation of pigment. In Arnon's method, the researcher physically grinds the leaf tissue using a mortar and pestle, adding 80. Beyond pigments, extracting nucleic acids serves as a universal compositional proxy that works across all biological kingdoms. The premise is direct. A viable cell is metabolically active, transcribing genes. A higher quantity of cellular DNA and RNA in a sample directly correlates to a higher total cell count. Using a nanodrop instrument, researchers measure the total concentration of the extracted genetic material via light absorbance at 260 nanometers. They confirm purity by measuring the 260/280 nanometer ratio. 1.8 indicates pure DNA, and 2.0 indicates pure RNA. When rigid cell walls prevent physical isolation or liquid suspension, isolating and measuring the tissue's core biochemical blueprint provides a quantifiable measure of growth. This matrix organizes our methodologies, revealing the selection framework of quantitative biology. Precision requires matching the distinct biological question to the correct proxy. If a process only demands the total yield of a culture, you rely on the physics of light scattering and the Beer-Lambert Law, or the measurement of dry weight. If the protocol dictates you confirm active life, you leverage biochemistry, utilizing dyes that only react to the metabolic activity of mitochondrial dehydrogenase. And if you are measuring complex, differentiated tissues, you bypass the macrostructure entirely, extracting and quantifying the foundational nucleic acids or photosynthetic pigments. Bioengineering scales up by moving past the limitations of the human eye. We achieve precision by selecting the exact physical or chemical lens necessary to make the biological system measurable.